a postdoctoral researcher. He finished his PhD in 2024, and during that time, he became quite well traveled. He spent some time in Sweden in the lab that I'm currently in now, but unfortunately, we didn't overlap in the lab, as well as some time at the University of Minnesota as well. And so he's going to talk a bit about his research in senescence um, and his career journey so far. So with that, Osama, I'll hand over to you, and thank you again for joining us today.
Thank you, Samir, and thank you, Florencia, for this kind words. I'm glad and happy to be here, and thank you for the invitation to share my story and how I came across senescence and my PhD time and journey. So here, we'll see if this is going to work. Okay. So I will walk you through my career journey and then a brief introduction about senescence and ovarian aging. Also, specifically, I'm talking about my PhD work that is about SMAD3 and senescence in aged ovaries. Also, speaking about few projects that I worked on during my time as a visiting PhD student in the US. Also, I will share my personal opinion about how my research experience was in Italy and Sweden, and currently, actually, I'm still in Italy and in the US. So, starting by how I started, and I'm actually from Libya, the city called Misurata, and that's basically I got my bachelor degree in another city in the Far East called Tobruk, and then I moved to Misurata and I worked as lab technician there and then I worked as embryologist at the Al Amal Hospital in Misurata. Following this, I was very fascinated by embryology and infertility. I was like, okay, I want to have a master's and I would like to know more about research. And interestingly, I found an opportunity to go to Italy through a MIEC grant or MIEC grant, which is funded by the Ministry of Affairs in Italy. So I had the chance to go to a small city called Teramo in Abruzzo. I know this is maybe most of you haven't heard of it, probably Rome or Napoli, but believe me, this region has its own charm. So I can show you a few things about Abruzzo that probably you never heard about. We have mountains, Gran Sasso. We have this very interesting thing that Trabocchi, which is a place for fishing, but now they're turned to be restaurants. Also, we have the National Park Flumayella. So hopefully we can hold one of the yICSA in the future here, and I can convince you that this is a great place to come. Yeah, so basically, as I said, I did my master's then in Teramo in reproductive biotechnologies. Following this, at the same region, I moved to University of Gabriele D'Annunzio, Chieti, Pescara, which is where I am being now as a postdoc. I worked under the supervision of Professor Antonia Patruno. And during this time, during my PhD, I have worked with her on a few papers, but also I did first visiting experience at Karolinska Institute, which was under Federico Petrocola. And I'm actually really thankful for Federico because I haven't known nothing about senescence until I joined his lab. So was a very fundamental for me to go into senescence and spend this time and from experts in this field. So this was very fundamental. However, after that, I also again went to US at the University of Minnesota under Paul Robbins' lab, and this is where I'm going to share most of the research that I have carried out there. So here, most of the yICSA or senescence people know this, but I'm going to introduce this for the rest of the audience. So as we know, in aging, we have certain factors that could happen that can lead to aging phenotype in general. We have chronic inflammation, for example, mitochondrial dysfunction, telomere attrition, which is basically like what happens to the cells when they stop division due to the attrition of the telomeres. But also, we are focusing on cellular senescence, and these factors actually, or hallmarks, are actually interconnected. For example, senescent cells, they have SASP that can be considered as inflammation. They have mitochondrial dysfunction. They also have some epigenetic alteration. So that's why we are focusing on cellular senescence. Again, a brief introduction about senescence. Senescence could happen or be induced by intrinsic factors like replicative stress or oncogene activation or DNA damage. Also, it could be extrinsic by UV light or radiation. There we have few markers of senescence. For example, we have P53, P21. These are related to cell cycle. They lead to the inhibition of cell cycle enzymes and cell cycle arrest. Also, we have an increase in C-beta-gal, which is the lysosomal activity of senescent cells. Also, the lamin D1, which is a protein in the envelope of the senescent cells that can be changed or let's say decreased. And then we have a key feature of senescence, which is SASP. That's senescence-associated secretory phenotype that can lead to, by its own role, depends on the context. So if it's physiological, it could be beneficial, like tumor suppression, wound healing. And if it's basically deleterious, we can have carcinogenesis or senescence and reinforcement or chronic inflammation. So here, basically, I started working on ovarian aging, and that's I'm going to also introduce you to the ovary and what's about it. So here we have that ovary actually, interestingly, is the first organ to age, and that would be very interesting to study. And if you have probably understand why this is happening or how to delay this, probably the intervention can work also on aging itself. So what happens during aging is that we have different changes that could happen. First, in age ovaries, we have an increase in young ovaries, we have increased estrogen progesterone, and we have an intact ovary, so follicles, after follicles, secondary follicles, and a normal extracellular matrix. While in aged ovaries, we have atretic follicles, lower estrogen progesterone, and more fibrosis. Following this, my project actually started from a collaboration with Yusin Su at Columbia University, that she identified a non-coding variant in the region of SMAD3 that is linked to longevity. And the hypothesis is that when you have less SMAD3, that's favorable of longevity, and with more, then you have disease and aging. And I will let you know here what's SMAD3 pathway. So we have activation of TGF-beta receptor that can lead to phosphorylation of PSMAD 2 and 3, and then it translocates to the nucleus. One thing that is important to mention here that is actually upstream of P21, which is a well-known marker of senescence. In addition to that, so knock-in of SMAD3 centenarian variants actually led to a decrease in SMAD3 in a cell-specific manner. So we have endothelial cells here and hematopoietic stem cells. However, also the centenarians variant here led to a decrease in senescence markers as well. So why SMAD3? SMAD3, as I mentioned, was linked to centenarians, but also actually it's linked to collagen deposition and changes in external metrics. So we were thinking what about the context of ovarian aging and fibrosis and possibly other organs. So here we started by evaluation the levels of PSMAD3 in senescent cells. As you can see here, there's an increase PSMAD3. And then we went to in vivo models, and here I'm gonna tell you about this model, ERC1 minus delta mice. And these mice have this enzyme that is endonuclease enzyme that is inhibited. So this enzyme works by excision repair and double-strand break repair and interstrand cross-link repair. So these mice accumulate DNA more than regular mice, and therefore they have accelerated aging. And by 25 weeks, they already at their almost like 3 years mouse. So this mouse is useful because by 16 weeks, they already exhibit aging phenotype. And we used also these mice in the next slides. So here we have looked at PSMA3 levels in liver and kidney and lung, and we can see that it's increasing in liver, in also the wild type and the ERC1 minus, the prodroid mice, also in kidney, but only in the prodroid mice, while in lung there's no changes. Again, in ovaries, we see here that there is an exponential increase in pSMAD3, as you can see here, but a decrease in its inhibitor. So SMAD7 actually is an inhibitor of SMAD3. That's confirming that pSMAD3 actually is increasing. This could be a target for aging. Again, P21, we have an exponential increase in P21 at 24 months, also in P16 in different mouse age and in ovaries. Again, so here what we wanted to see is that by inhibiting SMAD3, would we see a senolytic effect? So just I will go through what's the senolytic and xenomorphic. Senolytic means that the drug or cells, senescent cells, could be eliminated selectively without affecting non-senescent cells. While xenomorphic means that decreasing certain senescence marker without inducing cell death. So here what we see is that using siRNA, SMAD3 siRNA, there is a decrease in senescence in cell number, which is we used here C12 FDG that is linked to the SA beta-gal activity, and it's green. So we have here less green and less number of cells. And then we used the drug CYS3, which is a pharmacological inhibitor of SMAD3 and different cell models. So we had MR90, human fibroblast, and also wild-type MEPS. And as you can see here, we have a senolytic effect in all these different models. Again, heterozygosity in SMAD3. So in this progeroid mice, we have seen an improvement in symptoms, in aging symptom scores, like dystonia, kyphosis, and there is an improvement. Also, a decrease in certain senescence markers in P16, P21, and liver, and lung, and kidney. And also in ovary, we seen similar effect, like CXL and also P21. Also, this is in ovarian tissue. Using CYS3, the pharmacological drug, to inhibit SMAD3. This is in ovary. Similar results also we have a decrease in P21 and certain SASP, so IL-6, TNF-alpha, and CP1, and also few fibrosis markers like collagen 4A1, MMP12, and telomerase. And following this, we wanted to know what's happening in this ovaries after the inhibition of SMAD3. So we did an RNA-seq in this mouse model by inhibiting. We have an inhibition of SMAD3 in this progeroid mice. We have at the beginning an upregulation of oxidative phosphorylation, also DNA repair, and the inhibition of IL-6 and STAT3, also TNF-alpha, which would mean that there is an inhibition of inflammatory response. Another paper that could be a bit old, but actually going to our direction, is that when in white adipose tissue, there is an improvement in mitochondrial biogenesis by MitoTracker, and also improvement in respiration. And now we are currently evaluating the effect of inhibition SMAD3 on mitochondria in our hands. So conclusion, pSMAD3 increase in senescence in human fibroblasts and aged ovaries. When inhibiting SMAD3 by genetically or pharmacologically, we reduce senescence in vitro. Also, SMAD3 heterozygosity decreased SASP and senescence in mouse in ERC minus delta mouse model. SMAD3 is upregulated in aged ovaries and P21 and P16. And CYS3 administration in vivo decreased senescence in aged ovaries. And as I said, by this RNA-seq data, we have an upregulation of oxidative phosphorylation, which is mitochondria, and also SASP or inflammatory response. For now, I will switch, not the topic, but I will start talking about some papers and projects that I worked on while I was in the US. So by starting by speaking about fucoidins, and fucoidins are extracts from seaweeds, so algae, and they are known for their activity to activate SIRT6, and this was a good start for us to start and see if they also can exhibit senolytic effect. So here we screened different species and types of fucoidins, and what we came with was 2 hits. First one is fucoidin MP, and the second one is V. MP is senolytic, so as you can see here, we have less number of cells. And if V is actually senolytic, which is xenomorphic, sorry, it means that the cells are alive, but they have less C12 FDG, so they have less green, as you can see here. After that, we evaluated their effect in vivo, and here we used this mice, and this is fucoidan FV, that their effect was pronounced in kidney and lung and quad. This is our real-time PCR, and this is RP16, P21, and different SAS, TNF-alpha, 9X6. Also, again, here the MP, similar results, kidney and quad. Following this natural age mice model, we tested this in ARC minus delta, so accelerated age model, and we have a better response in this mice. Here, as you can see, that in all tissues, we have a down regulation. And also, not to forget to mention that we have also an improvement in aging symptom score. Again, we have done RNA-seq to see what the mechanism of fucoidans and what's really happening. So we have here seen that there is an upregulation of DNA repair mechanism by fucoidan. Also, there is a decrease of Sin-Mayo, both panel genes. Also, we have the DNA damage response that is also increased and cell cycle. And following this, what we have done, of course, I'm not showing all data, I'm just showing few data. And if you're interested, you could just go and look more into data that is on bioRxiv right now. So here, again, we wanted to see if this effect is mediated by SIRT6. And one of the activities that we can check is the deacetylase activity. And we can see that there is an increase after treatment of fucoidan. Also, the self SIRT6 at the ADP reconciliation does increase with fucoidan. Following this, we tried, this is a very interesting way of measuring non-homologous in-join repair efficiency by using a plasmid yICSA that can lead to DNA excisions that can be seen here by the GFP. So yeah, when you have more GFP, means that more DNA efficiency, DNA repair efficiency. And this mCherry is actually just the transfection efficiency. And here what you can appreciate is that treating with fucoidan, we have a better DNA repair. And when we inhibited SIRT6 with this RNA, we have a decrease in this ability of DNA repair. And eventually, what we've done to confirm this further, we have done gamma-H2X, which is a marker of DNA damage. And as you can see here, treating with wild-type MEFs with fucoidan, we have a different concentration. You have a decrease in DNA damage. And after using the SIRT6 knockout MEFs, we have that we cannot have the same effect. And we asked also, does the senolytic effect could be affected by this SIRT6 knockout? And here we can see that there's a nice decline in C12 FDG positive cells, which is mean a senolytic activity, while SIRT6 knockout partially rescue that effect. And with that, so this is basically the paper about fucoidan. And then this is the next paper that I worked on was that we worked on developing a novel flavonoids senolytics that we use different flavonoids as a template for one of them, for example, Pescara, and we identified two senolytics here that they're like flavonoid analogs. Again, also we tested this two flavonoid analogs. We see here that in kidney, are the two actually analogs, and you can see here Pescara next to them, so you can compare the effect. And here we can see that they're more potent and they have better effects. Also, they're structurally more based on their structure, they're more bioavailable, orally bioavailable, and absorption. So here, yeah, as I said, kidney, lung, and brain, and gastric muscle, and also spleen. that we have a really pronounced effect on senescence marker by real-time PCR. In one of the analogs was tested in ARC1 minus delta, the progeroid mice. We have seen just a few, based on their age, for example, ataxia, or at the beginning of their lifespan in dystonia, but we haven't seen any significant change in overall health. However, as I said, I'm not showing all the data, but we have done prediction of direct molecular target of this two flavonoid analogs. And the first neighbor targets were PARP1, BCL2, and CDK2. And following this, what we did is that flexible molecular docking to see if they really can be targeting these targets. So here you have on the right our positive control of PARP1, and these are the two drugs. And here we have BCL-XL, the two positive controls, and these our two drugs, and also CDK2. And right now, actually, we have also new data that by enzymatic activity and by assay that, but I'm not showing this data now here. So the take-home message is that proliferating cells have stable levels of CDK2 and PARP1 and BCL. They have lower, and then after, when you have a gene toxic stress, you have an increase in DNA damage, which is basically PARP1 and BCL2 as well, anti-apoptotic process. And by using this flavonoid, you target them, and you have then a selective apoptosis in these senescent cells. This is the third work that I'm sharing with you as well. It's also a screening of fatty acid, PUFAs, so polyunsaturated fatty acid, that we tested few of them, and we identified ESA acid and ESA methyl ester. So the difference is that the esterification, and we have tested this in different cell models, MEPS and MR90, also HUVEC, so endothelial cells, and we seen, as you can see, you can appreciate that there is a senolytic effect in all these models. Again, also, as always, testing them in vivo. Here we can see that in the difference is that here the age of the mouse is 20, and here is 31, pretty old. And we have seen, based on the drug, so here is an ESA methyl ester is more potent than ESA acid in heart, in liver as well, and in brain, for example, both could attenuate or decrease senescence in these tissues. Again, also in old mice, different responses, depends on the organ. So we have the kidney and also the lung. We have seen some effects of ESA methyl ester. And also in the progeroid mice, similar data. We then followed by again, another test in ARC1 minus delta. There is a certain improvement in the middle health span of this mice. And with histological analysis in liver and kidney, we can see that there is a decrease in SA beta-gal in liver, kidney, a decrease in DNA damage as well, and an improvement in Ki-67 in these cells, and also the iron deposition in this two treated organs, liver and kidney. So to understand better how this senolytic effect is happening, and this is basically, we used on the left here two inhibitors of metabolic targets of ESA to see if this could be the reason why we have this senolytic effect. But after the treatment with these inhibitors could not rescue cells from cell death. Going to the basic, as most of senolytic works through apoptosis, we used pan-caspase inhibitors, apoptosis inhibitors, and also necrosis, and still both of them could not rescue cells from death. Interestingly, we used iron chelator, apoptosis inhibitor, and also a lipid peroxidation inhibitor. We see here that all of them could rescue this cell death, meaning that this is probably how ESA is actually inducing cell death in these cells. following this, as here, we can tell that there is this. So here, just to make it easy, so we have non-senescent, senescence, and then yICSA, and then yICSA with a ferroptosis inhibitor, just to confirm that this is through this. So here, from this, you can see that this is for ROS, so reactive oxygen species, and this is for ferro-orange for ROS, for iron, and this is for lipid peroxidation. So senescent cells already shows that they have, but per se, they have higher, as you can see here, ROS and iron. Also, they have higher lipid peroxidation. And when you put yICSA, you have actually even higher, basically, iron and also ROS and lipid peroxidation. But when you treat them with, so you can follow the quantification here, when you treat them with the ferroptosis inhibitor, you have basically, you abrogated this effect of yICSA, yICSA here. So here confirming that this effect actually is due the ferroptosis process. And then we followed this by in silico prediction pathway to see if this, how these two ferroptosis fatty acid are different from known senolytics. And here you can see that this, and on the right side, we have well-known ferroptosis inducers. So just as a positive control. And if you, when you compare this ferroptosis induction in these two our drugs, in other senolytics, they're not targeting ferroptosis. Plus, these two, they're not targeting well-known conserved senolytic traditional target. For example, cell cycle, there is no target, there is no effect. P53, there is no effect. So these two senolytics, they have their own specific targeting, target by targeting ferroptosis genes. And based on the structurally probable targets of these two drugs, we identified two possible targets that they are the PPAR-G and ALOX5, and also here the same ALOX5 and PPAR-G and ALOX15. And I'm not showing the data, but then we did another analysis and we identified that the best target is ALOX15, but I will see here if I can show you. Yeah, after that, actually, I'm showing you here the lipidomic analysis. That basically what we can see here is that the senescent cells have more PUFAs substrate that's upregulated in general, but also after the treatment of yICSA methyl ester, we have an increase of oxidative PUFAs. And this is important because what we think is that senescent cells have more PUFAs substrate, and that's why they're more prone to lipid peroxidation. And what the yICSA does is that increasing more lipid peroxidation, exacerbating this effect and leading eventually to cell death by ferroptosis. And here, using different inhibitors of key targets of ferroptosis as well, again, here SACL4, we use SACL4, also LPCAT3, also ALOX15, which is our target as we predicted. As you can see here, when you treat this the cells with this SACL4 target, you can see here that when you treat, there is no effect of yICSA actually. So here, as you can see here, this yICSA alone, there's cell death, but then here there's nothing similar to all of them. So this means that this yICSA actually effect mediated through these three targets in ferroptosis. And again, another enzymatic activity, I say that we have to verify it's really 15 LOX activated. And here you can see that with this is an inhibitor, and with the yICSA, we have an increase of this activity. So confirming this is the potential target. So the key home message here is that healthy cells have low iron and low ROS, and they store their lipids, they store their tags as in lipid droplets, while senescent cells that they already have higher iron and higher ROS. So due to that interaction can lead to ROS and oxidation. And by this, basically yICSA through ALX15, which is an enzymatic reaction, can be like what we can call here adding fuel to the fire. And basically adding by this incorporating to the plasma membrane, leading eventually to cell death. So this is the effect of yICSA based on how the cell has of lipid peroxidation and iron and ROS. And as the senescent cells prove that they have higher, then that's why they are selectively eliminated by this. And then here, I'm not sharing with you the data, but I also had the pleasure to collaborate with Maria Iconumbulo. And here she discovered a cyanotoxin that is a marine toxin that can form pores in the senescent cells membrane and changing the lipid binding specificity and ion balance, also changing the lipidome. So I encourage you to have a look on this nice work. Then now I'll switch the gears to my experience in Italy and US as how I felt from my opinion. And of course, this is personal. So here, in my opinion, Italy, it's a good place to understand and learn basic biology, but you wouldn't probably, it's also lab dependent and you wouldn't probably get into cutting edge and more sophisticated techniques. Also, if you're looking for aging research, it's all usually embedded in other fields like cancer, metabolomics. So that's also, there's no real aging research course here. Grants, if you're looking for grants, that's also often short-term and underpowered. That's also something here. And fewer lab working on senescence specifically. But that said, I would say this is a personal, and also, of course, there are many labs that they have probably the techniques, the technology, they have the funding. Probably if you go to this lab, you will have a different experience. In Sweden, it's also the same thing. So I think your lab work on senescence, but I would say they have longer grant duration. And in my opinion, also the research, as always, it depends on the lab and the PI, but they have more collaborative environment. And the collaboration is a part of the environment. And then in the US, so US is basically the hub for senescence and neuroscience. So they have a really huge funding, and they have even the NIA, National Institute on Aging, which is focusing on this. But sometimes it gets too high, and you have to push out to publish and big stories. So it's a bit fast-paced, I would say. But it can give you a high impact academic path. But also what I like about US is that you have multi-lab consortia, for example, Senet, which is basically they're trying to characterize senescence in different organs in human and mice using really amazing techniques like single cell RNA-seq or spatial omics. So as I said, this is personal, and I think still it depends on the lab, the university, and still, I think it's more personal rather than generalization. So I cannot really generalize here, but this is my own experience. So with this, I would like to thank Paul Robbins who hosted me to conduct all this work, and also my professor, Antonia Patruno. She gave me this opportunity to go abroad. And of course, the postdoc, he was a postdoc. Now he's assistant professor in University of Georgia, Athens. I'm really thankful also to Lei and to everyone who has listened to this. And thank you, guys.
Great. That was amazing, Osama. Thank you very much. Well done. So we have some time now to move to a Q&A session based on the talk. We have a couple that were submitted during the registration, but if anyone else has any questions, having listened to the talk, please either raise your hand or turn off your mic, or put your question in the chat. In the meantime, I can start with one of the questions that were submitted, and this is by Fabio Pietrosanti, and I don't think they're in the chat. No. So the question is, in the context of fertility, What's the most promising expectation of treatments for aged women in the context of senescence?
Okay, that's a very interesting question, actually. As far as I know, so right now, the only, I would say, intervention that has been used as intervention, of course, it's rapamycin, so mTOR inhibitors. And now, actually, there is a clinical trial that is ongoing at Columbia University for women that are aged between 40 and 45 to see if we can see actually, because in mice, we saw that the rapamycin could extend the fertility lifespan in women. So that's, I would say, the only intervention that now have shown in vivo in mice, and right now it's being tested. And then, of course, I don't know if there's other interventions that could improve the fertility, but usually I think they go hands by hands with caloric restriction probably and exercise. That's always would be, I think, a useful thing.
Has anyone looked at this is just my own question following up that clearing like with a genetic model of just clearing senescent cells in the ovary, with a mouse model like the ink attack or something.
Yeah, that's actually, no, I haven't seen any paper. I know that some people would like to do that.
Mm.
Not yet.
Yeah, that would be cool.
Not yet. Probably, as you know, most of the mouse models, I think they involve tamoxifen, which is very toxic and could already have its own negative effect on ovaries. So unless you have a clean model that wouldn't have toxicity, but I don't know how you can induce removing. I mean, I don't know if ink attack, for example, by giving the AP is if it's toxic. I have no idea. But
Yeah,
this question probably need to be asked to the people who worked on this.
yeah,
Yeah,
absolutely.
but it's very, 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 very cool, very cool thoughts. Yeah, it's true.
Yeah.
It's very makes sense.
Thank you. No, thank you. We have another question also from the registration by Rabia Tabusun, which is asking about the mobility period and what sort of time is the best or which is the best option to go. So I don't know whether that refers to which place from your experience or what stage of your career is best. Maybe you can
Okay.
comment a bit on that.
Well, in my opinion, I think the best is during your PhD and probably, I mean, of course, I don't think there's a best based on that, but depends on what you're doing right now or where you're at.
Mm,
So if you have everything that you have access to everything, and then maybe you have an interest in certain expertise, and this expertise are existing in country A or B, then you go for that. And basically, a good thing in Italy, they give you really, you can even stay for a year and a half, which is amazing. And that's
yeah.
what I did basically. Um, about the place, I mean, for postdoc, I think maybe, for example, Sweden or US. For PhD, I would say Italy. It's going to be good to do a
Mm,
PhD. But yes, the question is too broad, so I don't know what.
yeah, yeah, of course. Um, No, that's really helpful. Thank you very much. Those were the two questions I had in the chat. So I would like to open it to the audience if anyone has any comments or questions about the science that Osama showed, or to get his expertise. We have a comment now from Himanshu. Would you like me to read it, or would you like to? You're welcome to.
Well, yeah, yeah, go ahead, go ahead.
I can read as it's in the chat.
Yeah,
The question is from Himanshu Sharma. Is studying combinations of senolytic drugs of different classes worth it?
absolutely, yeah. I mean, I think it's just people they don't do it, but probably because it's I don't know, maybe it's costly
Yeah.
because you need to try in order to say this is the effect, you need to try together the drugs
Mm.
alone and then the combination. And this
Yeah.
would probably require more mice or more, and that's most of the, I mean, I think PIs or labs don't go for the combination, but I think it's really useful. I mean, of course, you can show this works probably in vitro, but
Mm.
then you still need to verify this in vivo. And to doing this, it's usually more expensive and costly. So I think that's why most of they don't go for it. But of course, I mean, I think it's really cool and it's worth doing it.
I 100% agree, and I suppose on the other side as well, it's important the context at which you're studying, because I guess we could look at aging as a whole, but
Yeah,
the specific tissue we're looking at, the cell types that are becoming senescent and
exactly,
what pathways are
exactly,
vulnerable to, and then um, and the side effects, I suppose, of the multiple drugs that you can use too.
exactly. I cannot agree more. Yeah.
Yeah, it's a great point. Um, but yeah, if anyone else has any other questions, feel free to ask. Otherwise, yeah, we do have one. This one is from Christina Battaglia. The question is,
Mhm.
do you think that organoids can be good in vitro models to study senescence?
And that's a very, um, for sure. I think already organoids are like a step further from usual 2D or any other in vitro models. So I think, yeah, of course. But from my experience, I haven't worked on that, but I think it would be cool to work. I think, yeah, it will be useful if you have let's say the tools to study this and see. We never know, but it's I think it's an area to worth exploring. Do you have any idea, Samir? Have you heard about anyone working on or with organoids with plasmids? For
People
as far as I
definitely
know,
do. Yeah, but in our lab, we're trying to get some lung organoids set up, for example, but it does take some time. You need some
yeah,
technical and theoretical expertise.
yeah,
We collaborated with some people for this, but I agree 100%. Um, you get more of a real microenvironment, and especially with human samples, right?
yes,
Because,
yeah.
yeah, yeah, I agree. Florencia has her hand raised. Would you like to ask a question?
Osama,
No tough
amazing talk,
questions,
amazing
Florencia.
results.
Okay.
Uh, I was wondering, maybe you mentioned this in your talk, but is it known which particular ovarian cell type is affected during aging in regarding the phosphosmalt phosphorylation, like resident macrophages, endothelial cells,
Yeah.
granulosa? Is it something no?
That's a very, that's a question that I've been asking during my PhD all the time. So, and I also tried to uh, figure this out by doing some. And right now, actually, it's ongoing with Senate. They are analyzing ovarian tissues from human and mice. Um, right now, the only thing from the literature is that they have seen from different age, for example, like because as you know, mice are a bit different. Plus, when the mouse is 23, it's hypothetically like post-menopause. So there are different stages that people looked into the ovaries. So when you look from nine, for example, 10 months, which is like probably it's the starting of the menopause, they have seen that there is a changes in certain subset, for example, T cells or other endothelial cells, while when you wait until the like time pass, there's another subset that's changing and probably could be senescence, P16 positive, for example. And also that's the tricky thing as well, always. How are we considering them senescence? Are they P16 positive, P21, DNA damaged? What's the main marker? And that's the main, I think, I would say, challenge in this
Mm.
characterization. But right now, I would like also, I'm just like you, Florence, I would like to know what cell is uh, going senescence first in ovaries and probably could lead to this. As you can see with age, yeah, we have more senescence cells, but which one? No idea. Okay.
Thank you.
Francesca has her hand raised.
Yes.
Please.
Ciao, Sama. Thank you very much for the talk. It was very nice, very well done. So congratulations.
Thanks.
And I have a quick question. So at the beginning of the talk, you mentioned about the data from Centenarians. Now, I'm not able to recall to remember properly.
Mhm.
But how? I mean, did you study, factors in the blood of these Centenarians, or it was organ? So how did you find then phosphosmad in that population? Can you remind me?
Oh, okay, yeah. So I would, if I'm not wrong, if I recall well, I think it was from blood,
Mm-hmm.
and it was basically they have done a technique called capture-seq, and to
Okay.
see and they focus more on TGF beta and certain pathways. So it's more targeted approach. It's not like a more wide range. And based on
Mm-hmm.
that, they saw that there is this certain non-coding variant that is inhibited. And by the way, it's inhibited through a transcription factor called ALK1. and this is the one that goes and inhibit. So it's mediated by this. The only caveat is that this is ILK1 is not conserved in mice. So we don't have the same binding site. So so we don't really, that's why we could not, I mean, the good way is that you take the centenarians variant and you put it into mice, but we couldn't because it doesn't exist.
Okay. I see. Okay. Thank you.
Thank you.
Great. So if there are no more questions, I think we will close the session. Thank you so much, Osama.
Thank you, Samir, and thank you, Florencia.
Yeah, and thank you for the people asking questions. Just before we close, I want to let people know that we will have another yICSA webinar on the 6th of March. So keep an eye out on our socials. We will share that information very shortly. So until then, we will see you all again soon.
Thank you, everyone, for attending. Ciao.
Ciao.
Thank you, Osama. Fantastic meeting.
Thanks. Thank you, Florencia. Thank you so much.
Have a great weekend. Bye-bye.
You too.
See
Bye.
you.